Hello there, and welcome to your Biology 101 lecture for this week. As you may know, each of these lectures will track with the Ivy Tech statewide 101 online biology class. And so what we'll be doing today is exploring some cool aspects of how chemistry relates to biology. And so you may be thinking, well, gee, I signed up for a biology class. Why do I need to learn about chemistry? And there's a couple reasons for that. One, if you think back to Chapter 1, you showed how the hierarchy of organization of biological organisms starts with atoms. Those atoms can be used to make molecules, and so on, until you build up to a whole organism. Secondly, chemistry has dramatic impacts on the functioning of biological systems. And we'll explore one example of that in depth today. So what we'll do is we'll start off with a PowerPoint presentation associated with Chapter 2, and then one with Chapter 3, and we'll try to highlight some cool aspects of these subjects as we move along. So, Chapter 2, we're going to start with the atom. And atoms are the fundamental unit of matter. They are obviously very, very small, and they have three important parts: protons, which have a positive charge, neutrons, which have no charge, and electrons, which have a negative charge. Protons exist in the atom's nucleus, as neutrons do as well, but electrons move around the nucleus at some distance from it. And so this here is kind of the planetary model of an atom. And what you have is a nucleus that has protons and neutrons, and then electrons that circle around that. However, this model isn't exactly accurate. These electrons do not really go around the nucleus in this way. Really, they're bouncing around the nucleus all the time, and they're more likely to be found toward the center and a little bit less likely to be found farther away. So you can really think about these electrons like a cloud around the protons and neutrons, or moving within a realm around the protons and neutrons. But we will talk about this using this model today, because you'll see how it's very valuable to your understanding of how these atoms actually interact with each other. Because when atoms interact with each other, that's where we get chemical reactions, and we can build molecules. An element is any substance that cannot be reduced to any simpler set of constituents through chemical means, and basically this is something you would find on the chemical or the periodic table of elements. So for instance, carbon, nitrogen, oxygen, helium, all these things are elements. Well, you can have a single atom of gold, for instance. And we've told you about protons, neutrons, and electrons. Sometimes we can mess with the number of protons the number of neutrons in an element. Protons really define what that atom is, what element that atom is. Neutrons don't really do that much. When we have extra neutrons, that's called an isotope, and all it really does is slightly change the mass of that atom, but it does not fundamentally change its behavior. Atoms of different molecules are going to interact with each other. And we're going to talk about this through the covalent bonding, and these atoms can link to another in a process of chemical bonding. We have covalent bonds and ionic bonds. We're going to mostly concentrate on the covalent bond where they share electrons. And we can kind of sum up how atoms are going to interact with each other to make molecules using a simple rule about energy state in an atom. And an atom is trying to achieve their lowest energy state, so to speak, by having a full valence electron shell. And that may not mean very much, but let's look at the board here and explain some of this. So, the goal of an atom is to have a full outer energy layer. And so, if you imagine we've got a nucleus here, and we wanted to start adding electrons around that nucleus, what we would do is the first layer holds 2 electrons. Let's change this to black, and this green is not showing up very well on here. There we go. First layer, 2 electrons. Then we're going to add more electrons to this. And when we do that, we had 1, 2, 3, 4, 5, 6, 7, and 8 to this layer. And then thereafter, if we added another layer, if we had one more electron, it's got to move to another layer, and we would add our 8 again. And we could keep moving on and doing that. Now, remember, it's the goal of this atom to have a full outer shell. And it can do this in one of two ways, or it can achieve this in one of two ways. The first way it can achieve this is through addition. So if I had 7 electrons in my outer shell, what I could do to get to a full outer shell might be add 1, and then I would get to 8. That shell would have 8. If I only had 1 electron in my outer shell, what I would do to get to a full shell is I could give up that 1. Because if I give up that 1, I drop down 1 layer, but I still have a full outer shell. And so this is how atoms are going to interact with each other. And we can have a range of electrons in the outer shell, 1, 2, 3, 4, 5, 6, 7, and 8. And so, when you have 8, you don't do anything. You're happy. The atom has 8 in its outer shell. It doesn't need to change. If you have 7, what you're going to do is you can add 1 very easily. If you have 6, you can add 2 very easily. As we get into 5, the easiest thing you can do is add 3, but sometimes you can give them. And over here, if you have 3, it's easiest for you to lose 3. We're going to skip 4 first. And over here, you lose 2. Over here, you lose 1. So each of these, by doing this with the electrons, will end up at a full valence shell, stable and happy. But then we've got this guy here with 4. And it was easy for these guys to add them. It was easy for these guys to subtract them. What do you think 4 is going to do? Well, the answer is it's really good at adding or subtracting to reach that stability. So what this means is that it has a lot of potential to interact with different atoms in different ways. Furthermore, by having 4, it gives it the opportunity to create lots of linkages. And so a atom with 4 valence electrons is going to be very versatile. And it's this versatility that leads us to the element of organic chemistry, or the chemistry of life, which is carbon. Carbon has 4 valence electrons, which means it can do tons of awesome stuff. And all the biological molecules that we see in the next chapter will be built on a carbon framework. If you've ever watched science fiction or something like that, you may have heard of the idea that scientists say, well, there might be silicon-based life out there. The reason why they say that is because silicon is just below carbon on the periodic table of elements, and it also has 4 valence electrons. We could have potentially been based on silicon. It happens that we're based on carbon. So that's kind of summing up a whole bunch of chemistry into a very simple rule. When these things form covalent bonds, I was talking about adding and subtracting, but where are they really going? And we're talking about sharing. And so what we mean by that is if we look at a water molecule, we've got an oxygen atom. And oxygen starts with 6 valence electrons, so it really wants to add 2 to that total. And so what it does is it shares electrons with a hydrogen atom so that one would get it to 7, another one would get it to 8, and then it's stable. Meanwhile, these hydrogen atoms, remember that first layer can only hold 2 electrons, are happy because they are also sharing an electron with the oxygen atom. And so this becomes a very stable molecule. Water is very stable because the oxygen gets 8 and each of the hydrogen atoms get 2. You'll notice that we've got a lot of positive and negative charges floating around here. And this means that a molecule can be polar, or meaning that there's a difference in electrical charge at one end compared to the other. And a polar molecule simply means electrons are oriented toward one end and protons are located toward the other end, such that it actually acts a little bit like a magnet. Right? Magnets have a negative side and a positive side, and these will actually behave a lot like a magnet in what's known as hydrogen bonding, where the positive parts of one water molecule line up with the negative parts of another to create this linkage between the water molecules known as hydrogen bonding. Hydrogen bonding is fairly weak. It's not like the ionic or covalent bonding we were talking about before, but it does some things. So for instance, have you ever seen the insects that can stand on the surface of water? They can do that because of the cohesion of water molecules to each other, which is in part related to hydrogen bonding. And so this also, by water molecules wanting to stick to each other, allows water to be pulled through plants during transpiration, a part of photosynthesis. And you may have actually seen this in some of your labs before, and I think you'll see it in one of the labs later in the semester. How do you measure a column of water in a graduated cylinder? Well, the water is going to dip, and I'm going to exaggerate this dip to show you, and you measure it from the bottom of the dip. Why doesn't it just lay flat like that? Why wouldn't it have this dip? Well, two things. One, there's adhesion of water molecules to the side of the container. They stick to the sides a little bit, but then, because of the cohesion of water, the water sticks to each other. And so you've got water molecules stuck to the side, and so they can stick to each other, and it follows this trend because that sticking of water molecules is fighting gravity. So molecules have three-dimensional structures, and a three-dimensional molecular shape is important in biology because it determines the capacity that molecules have to bind with one another. And so here's the example your book gives you of you smelling bread. And so inside, back here in your nose, there are receptors that can bind to specific molecules. And bread has specific molecules, and when you bake it, those molecules become more volatile, or in other words, they want to jump off the bread, right? You can smell baking bread very well, but you can't smell bread that was baked a long time ago as well, because hot bread, lots of kinetic energy, lots of molecules jumping off. Those molecules jump off the bread, and they bind to their specific receptors. And so they give you this example of a triangle over here, like that. This fits perfectly in there like a lock and key. A circle would not fit in there, so you wouldn't have the same sensation. A square would not fit in there. The shape of the molecules will be different to fit in there. When this binds, it sends a signal to your brain. Your brain says, oh, I can smell that, and it detects that signal, and it interprets it or integrates it as the smell of bread. And so what I'd like to do now is take a little bit of time to give you an example of how chemistry really influences the biological world. And we'll use our kind of molecule receptor idea here to talk about a few examples of how estrogens can influence the biological world and some cool examples of that. So let's start off with estrogens in the uterus. Imagine you had a mouse, and this is its uterus, and inside that uterus you had one male and 7 females. Mice can give birth to lots of babies. When males are developing, they produce male hormones. When females are developing, they produce female hormones. But because these are all squished in there together, some of the hormones produced by this one can sneak in and affect this one. And so a male mouse that is born with 7 females is less masculine than a male mouse that is born from a uterus that contains, say, 8 males. Let's use the other extreme. And by less masculine, I mean it will display less aggression. They may be a little bit smaller. They will have less of those secondary sexual characteristics that we associate with males. And so we have an example there of estrogen inside a body affecting something. However, estrogens can also occur outside living organisms. And this is because of a very interesting quirk of the shape of molecules that we were talking about before. And if you remember, we had our receptor, and it looked like this, and there was a triangle here, and that fit in there. Well, have you ever been told not to microwave plastics and then eat or drink substances that came from them? And you ever heard, oh, because it's bad for you. Well, the reason is because those chemicals that slough off the plastic are environmental estrogens. They are chemicals that are chemically similar to estrogen and can bind to the receptors for estrogen. So, for example, this triangle fits in here, but we can put whatever else we want on the back end of that molecule, and it can still fit there. And so chemicals can mimic each other in nature. And so, for instance, there was somebody who was telling me one time about an experiment they were doing involving estrogen in rats. And they were trying to look at the reproductive biology of rats with high estrogen levels and low estrogen levels. And so what they did was they injected one group of rats with estrogen, and they had one group of rats that they injected with saline. Because they couldn't just not inject it. They had to go through the same procedure, but the injection was basically a blank. It was nothing, like a placebo. And in her experiments, she was finding that the reproductive biology, I think she was measuring health of offspring, was similar between the two groups. And that was that didn't fit with her hypothesis. And so, if you remember the scientific method, when you get to the end, you go back to the beginning. You ask new questions. You try to figure out what's going on. We just don't say, oh, I was wrong, and abandon it. So she explored it further. And what she found out was, well, what she wondered was, are my injections doing anything? So she went and she measured the estrogen levels inside the rats that were injected and inside the rats that were not injected. And what she found was that both groups of rats had really high estrogen levels. And she wondered what this was for, why this was happening. And she finally traced it to a source. And that source was she had decided to feed the rats dog food because it was easily available and easy to feed them. And a lot of people have this idea that dog food is full of like animal parts and stuff like that. But more often than not, the protein that you get from the dog food comes from soy. And soy is full of a type of environmental estrogen known as phytoestrogens. They are plant chemicals that mimic estrogen enough such that they can bind to estrogen receptors. And so what she found in her experiment was the effect of these animals eating so much soy with so many phytoestrogens was swamping out the effect of her injection. And after she discovered this and she changed the rat's diet, she was able to successfully test her hypotheses and move on. So those are examples, inside a uterus, something that can get into your food from microwaving something. Keep in mind, there's probably not enough in there to actually do anything to you if you eat food that was microwaved in plastic. Let's take it to an example of how these might affect things globally. People were looking around certain farms and finding that frog populations were declining. And they kind of said, well, why is this going on? There used to be so many frogs. Frog populations are going down. They're looking for all sorts of different reasons. And there are many reasons out there. But one of the reasons that they settled on was the idea that this could be caused by a herbicide called atrazine. When you apply chemicals to a farm, they just don't stay where they are. They move around. And so when rain comes, runoff pushes them into water bodies. And inside these water bodies, you have developing frogs. And frogs are mushy, and they're surrounded by the environment they're in. So if there's something in that water, they're coming into contact with it. And when they looked at these frog populations, what they found was that the male frogs were having all sorts of abnormalities. So for instance, inside male frogs, you would find reduced testes. Inside some male frogs, they even found they were producing eggs. Male frogs would not sing as much. Male frogs would act like females. And as it turns out, as you might expect, atrazine was an environmental estrogen. And this was influencing the male frogs to be less masculine, and therefore their reproduction was all messed up, and frog populations were declining. We can take this a step further into some research that's going on right now. And this is still not published yet. These are preliminary results. And what these researchers are finding is that we also, near places with large human populations, are finding male frogs that have the same problems like we found in the places that were using atrazine. And there were a lot of ideas for this. But there's one that somebody really hit on by comparing cities that had universities in them and cities that did not. So we've got two cities, let's say, each of which has 50,000 people. All right? But this city, a huge portion of the population is made up of university or college students, and this one is not. So we'll just put an X there. Keep in mind, none of this has actually been totally demonstrated yet. It's just an idea that somebody's researching. There's preliminary results that suggest it's true. What they were finding is that outside the wastewater treatment plants, at cities with universities, there was a great number of male frogs that had the problem we discussed. Outside the standard population, that number was less. So we have a high number here, let's put a plus, and a low number here. And the reason they think this might be the case, and the reason is that on college campuses, you have a great number of women using birth control, chemical birth control. In other words, the pill. What the pill is, it's a large dose of female hormones, including estrogen, that basically trick the reproductive cycle into continuing its cycle regularly each month. So this is a large extra dose of estrogen. And when you have a large extra amount of chemical in your body's going to eliminate that. And they eliminate it, and people eliminate these chemicals through their feces and urine. And so what happens is this leaves the dorms in the university, goes to the wastewater treatment plant, and those estrogens were ending up in the water with the frogs in those populations. Populations that have fewer individuals or less people using birth control have less estrogens in the water and potentially less frog deformities. And this comparison between universities and not universities is still being explored, but the idea that city populations could be influencing the frogs is pretty well established. So, that was a little bit of an aside, but I hope it gives you an appreciation how just one molecule and how the shape of that one molecule can influence a dramatic number of things. To the point where people taking birth control could be causing declines in frog populations because of one chemical moving through the environment. So, we talked about water a little before. Let's talk about it again real quick. Water has several qualities that have strongly affected life on Earth. A solution is a homogeneous mixture of two or more kinds of molecules, atoms or ions, and the compound dissolved in the solution is known as a solute. The compound doing the dissolving is known as the solvent. And so the easiest example of a solution is salt water. Salt water, in that example, the solvent is the water, and the solute is the salt. And we can kind of do this for any solution that you see. So for instance, if you make Kool-Aid, when you make Kool-Aid, the solvent is still water, and the solute is whatever the heck is inside Kool-Aid, whatever chemicals are in there. Sorry, solvent, water, solute, Kool-Aid. So water is really great because it's a powerful solvent and it has the ability to dissolve tons of compounds in a greater amount than any other liquid. Water also is important because water's solid form ice is less dense than its liquid form. And what density is, it's how many molecules are packed into this space. And so things that are less dense would have fewer molecules in a given region. And so the way this works out with ice is kind of weird. Most things, as you make them colder, become more dense. But with water, it is most dense at about 4 degrees Celsius, and it becomes less dense when it's ice. Ice actually has the molecules spread out a little bit farther than water. And you can tell that this is the case because ice floats in water. It's less dense, it's functionally lighter, so it goes to the surface. And so what this does is it gives us a effect in lakes. If we draw a lake, here's the lake. We have water in the lake. When that water freezes, because it's cold outside, it stays near the top. Because ice floats. If ice did not float, what could happen in small lakes was you would freeze some water, it would start to sink, it would slowly make everything colder until ice you were forming would sink to the bottom and this whole thing would freeze. A little bit more complicated than that, but you get the idea. But because the ice floats near the surface, the water becomes stratified, and you end up with 4 degrees Celsius at the bottom of the lake, and then the water in here is a temperature closer to 0 when it's ice. And you can tell lakes can be stratified with their temperature like this. If you've ever been swimming in a lake and you dive down and suddenly it gets really cold over a very sharp period, there's a thermocline there, or a quick change in temperature because of the stratification of the water. The other thing this does, the other effect this has on lakes, is it causes seasonal turnover. Because the ice is less dense, as you heat it up, it becomes more dense, and the water that was at the surface wants to go to the bottom. Well, that means the water that is at the bottom comes up to the surface, and you get this cycling of water inside a lake, and it actually moves nutrients that had all sunk to the bottom of the lake and deposits them again on the shore where lots of things are going to grow. Water also has the ability to absorb and retain heat, and because of this, the oceans act as a buffer for the Earth's temperature. And it may seem weird to think about buffering the temperature of the whole Earth, but let's look at this in the context of something that maybe applies to your life. Buffering the temperature inside your house. Think about if you had your house and it was fall. And so in fall, you kind of have cold nights and warmer days. What you could do if you were trying to keep your house, let's go with keeping your house warm, is during the day when it's warm, open up all your doors and windows and let your house absorb all that heat. When night comes, close all your doors and windows. It will cool down outside, but your house will have started out warmer. And so your house will slowly lose that heat through the night. But then the next morning, you can open up your doors again and let the heat in. And the idea here is that your house is buffering the temperature. It's resisting the change because it's stored a bunch of heat. And so you can actually save on your electrical or gas bills or whatever by just using this principle of buffering. And so the Earth does that in the same way. It heats up the water a lot, but that water stores the heat so that when it gets cold, it loses some of that. Acids and bases, we're not going to spend much time talking about these. If I told you I was going to put a drop of acid on a piece of paper, you would all say, oh, it's going to eat through it. And this is because acids and bases are basically reactive. They react with other things, and a lot of people have this idea that acid is bad and bases are not, but bases can be just as nasty as acids. For instance, in this example, you would obviously not want to drink battery acid, but likewise, you would not want to drink bleach, right? And that's because both strong bases and strong acids are very reactive. You can actually get chemical burns from bases just like you could acids. And just when you're working with the pH scale, keep in mind it is on a logarithmic scale. What that means is that a linear scale, for every change in 1, there would be a change in 1 over here. On the pH scale, what you actually have is for every change of 1 in pH, you get an increase in tenfold of its reactivity. So it's not a 1-to-1 ratio. And living things, most of them function best near a neutral pH. So for instance, the pH of your body is very, very close to 7, which is neutral. You're actually a little bit off. You're about 7.35 or 7.65, someplace in that range. All right. So let's go ahead and move on to the second chapter, chapter 3, the biological molecules. We've got to get rid of this guy. There we go. Get to that. Okay. And so this section starts with carbon. And we kind of already talked about this. Carbon is a central element to life because most biological molecules are built on a carbon framework. And carbon's outer shell has only 4 of the 8 electrons, which we talked about, for maximum stability. And so because it has 4, carbon is able to form a whole bunch of awesome covalent bonds with a wide variety of other atoms, including carbon atoms, to build these backbones for the biological molecules of life. We take carbon and then we add stuff to it, and those things are oftentimes known as functional groups. And what functional groups are just other atoms that we stick onto this carbon framework. And so, for example, you can take a whole bunch of carbons linked together, put an oxygen and hydrogen off of the side, and then you have an alcohol. And there's lots of different examples how we can add these functional groups to the sides. And so I'll show you the structure of a molecule again in a second. All right, let's get into our types of biological molecules, and we're going to start with carbohydrates. Carbohydrates are formed from building blocks or monomers of simple sugars such as glucose, and then we link a bunch of simple sugars together to make a polysaccharide or a carbohydrate. Monosaccharide is one sugar. Polysaccharide is many sugars. And they can make several things in the living world, such as starch, glycogen, glucose, cellulose, or chitin. And here we have an example of a starch in the form of a potato. And let's look at starches, potatoes, and how all this relates to the living world. And so, if you look at this figure in your book, it's kind of difficult to see on your screen, I know. We have a potato, okay? And what we're starting here is the story of how we make starches, of how starches are made, and how we can utilize those starches to get energy. And this is a story putting the world in the context of the carbon cycle. We'll talk about this today, and it will actually be a unifying theme that we see several times again throughout our lectures this semester. And so we start off with our potato, and what is that potato going to do? Well, that potato doesn't start as a potato. Let's actually take a step back. It starts as a seed. Let's start with the seed. And that seed is in the ground, and it's going to grow. And so it has some energy reserves inside that seed, and so it grows a little plant. And that plant has little roots. Okay? But how are we going to go from a little plant to a big plant that's making a potato? Well, we're going to do this through the process of photosynthesis. And as you all know, photosynthesis requires the sun. But there's more to it than that. Because you can't just turn energy into matter. This plant actually has to get the atoms that it uses to build itself from someplace. And a lot of people say, right away, it's going to get water and nutrients from the soil, and that's where it's going to get the things that it needs to grow. However, that is not the whole story. There's one very big, very important part of the story, and that's that we are building organic compounds that are based on carbon, and as this plant grows bigger, where does it get those from? Well, it gets them from the atmosphere. Because as you may have heard before, plants output oxygen, but what they actually bring in is CO2. So, what's happening is that plant is just grabbing that carbon. The plant brings in CO2, and using energy from the sun, it builds biological molecules such as a sugar. And all sugar typically is it's a carbons stuck together in a ring with those chemical bonds we were telling you about before. And you can usually have 5, or let's draw 6 here, just to keep it consistent. Carbons stuck together in a ring, that's a sugar. It's that simple. We grab the sugars from the CO2 in the atmosphere, put them to build more plant parts, we output oxygen. And so what's going on here is because of photosynthesis providing energy, we store some of that energy in the bonds of this molecule. And so the plant is trying to store energy. What it's trying to do is build a big potato down here so that it can save up energy in order to have a big reproductive event. But we don't let that happen. Because what we do is we come along, we steal this potato from this poor plant and eat it. The potato wasn't growing the potato because it wanted to feed you. It was growing it to store its own energy. But we're going to steal that energy because we're jerks. And so here we are over here, and we are going to eat this potato. And we are going to get energy from this potato by breaking up the carbohydrates. Because a carbohydrate, remember, it's a polysaccharide. It's a bunch of these sugar molecules stuck together. So the first thing we do is we break up the carbohydrate into simple sugars. And you can test this out on your own. You can test out the fact that carbohydrates are actually made of sugars if you do a little experiment with a saltine. What you can do is take a saltine, something you would not consider a sweet food, and put it in your mouth to eat it, but do not swallow it. Chew that saltine for 1.5 minutes, 2 minutes, and it will actually start to taste very, very sweet. And this is because enzymes, or chemicals in your mouth, break down that sugar, or break down those carbohydrates into the simple sugars. Those simple sugars can bind to your taste buds, and you can then taste them. And so you eat this, you break the bonds in these carbohydrates to make something known as ATP, which is going to be a universal energy molecule that you can do stuff. You basically transform the energy from the sugar into the ATP. But you've broken this up now. You've got a whole bunch of extra carbons around. What are you going to do with them? Well, we know that plants take in CO2 and emit oxygen. What you do, as you may know, is you breathe in oxygen and you emit CO2. And the reason for that is you've got a bunch of extra carbons left around. Not all of them are eliminated in your breathing. You know, obviously you defecate some of them. But a lot of them come out in CO2. And then because they're back out in the atmosphere, the plant can absorb them again. And it's how life functions. So we just talked about you eating starches. And we know starches are composed of simple sugars, which is why they are sweet. Or sorry, why you could do the saltine experiment. But if I was to survey all of you watching right now and ask you, how many of you like sugar? I'm guessing that almost every single one of you, actually I'm not guessing, I know almost every single one of you, yeah, I like sweet things. And most people like sweet things. Why do you think people like sweet things? Well, the answer is because your ancestors like sweet things. And we're going to hit on the topic of evolution here that we'll talk about in a little bit later in the semester. Evolution, another unifying theme of the course. So let's just move, let's get rid of this here for now. And let's talk about two cavemen. Caveman 1 and Caveman 2. Caveman 1 gets a positive, happy feeling from eating sugars. When they eat sugars, they feel good about it, they taste sweet, they like it. So they're encouraged to go out and eat lots of sugars. Caveman 2 does not have this. When they encounter sugars, they say, you know, I'm going to pass, they don't like them very much, not a positive experience. And so, what do you think is going to happen to these guys? Well, cavemen, life is not easy. They're hunting and gathering. And this one is going to end up consuming more calories because they like sugars. And so let's say a harsh winter comes around. Poor caveman number 2 is dead because he didn't get enough calories. Well, caveman 1 has lived because they did. And so the cavemen left alive are the ones that like sugars. And as you know, Homo sapiens are cavemen. We are related to cavemen, and we came from them. And so we came from a population of people that like sugar, and so we like sugar today. But what happens to you if you eat lots of sugars? Well, you end up with putting on lots of lipids. Lipids is another term for fats. And a lot of people have this idea that eating fat makes you fat. Like you eat fat and it directly becomes fat in your body. However, that's not exactly the case. Fats are very high in calories, but your body can take any of these biological molecules and convert them into fat. Why might somebody need fat? Well, remember our caveman over here? This caveman was eating lots of food, but one day he may have lots of food, but another day he might not. So when there's extra energy in abundance, animals store energy as fat. So this is him getting a big belly like I'm starting to develop here. And so he puts on fat, and then maybe winter's really hard. He loses that weight, but he had a form of stored energy. And this gets us to an interesting thing of the combination of the fact that we're driven to eat things that we like. We are driven to consume fatty foods, sugary foods that are high in calories. Why? Because remember, the cavemen that liked those things had higher fitness. However, what's happened today is it's very easy for us to get those foods. And this has led to obesity in the United States. Because what happens is people are hardwired to get pleasurable experiences from eating this food, so they eat a lot, but they can get so much of it that we pack on those pounds. And so part of the obesity problem is linked to the fact that we are hardwired due to evolution to want to eat a lot. And we now live in an environment where it's easy to get that food. But we all know that the obesity problem is not just linked to this hardwired thing. It's also because our lives are becoming increasingly sedentary. Or in other words, we're not moving around as much. And so the way this pertains to weight gain is one very simple rule. If you eat more calories than you burn, you will gain weight. If you burn more calories than you consume, you will lose weight. And this is actually the key to any successful diet. Even if the diets try to weave some story about how what they're doing is different, most of the time it comes down to these simple rules. And so, for instance, if you know somebody who wants to lose weight, they're going to change their diet and they might start exercising as well. They can do one or the other, they can do both. These diets, like I was saying, are all about meeting this equation of calorie consumption. And you may have heard of something like the Atkins diet. And the Atkins diet is all about eating more protein and less carbohydrates. Here's the tricky part to it. Remember, carbohydrates are broken down into sugars. Sugars, as we all know, calories, right? What that Atkins diet is doing is actually just getting you to eat less calories by eating less carbohydrates, and therefore, lower calorie intake, you can potentially lose weight. In other words, back to carbohydrates, eating a piece of bread is the same as eating a spoonful of sugar from a caloric standpoint, although the bread would have more nutrients and additional things inside there. So we kind of jumped into proteins there. And I want to talk about why proteins are cool for one very simple reason. Look at our lipids over here, or sorry, our carbohydrates. They are the same repeating unit over and over. Like, look, these nice lines of the same thing. Lipids also very similar, same repeating units over and over. There are some differences in lipids. They are a little bit more diverse than carbohydrates. But functionally, very similar. This is where proteins make a big showing in their diversity. If I gave you a box of Legos, and I said, hey, you know what? I would like you to build me something. And you're all excited because we're going to play with Legos in labs today. And you open up the box of Legos, and all you have is that one piece that looks like this. How many things can you build with that? And the answer is you can build sticks. Lots of sticks of different lengths. You can't do very much with it because you're only given one shape of Lego. But what if I came along and I gave you a second box of Legos and I asked you to build me something? And you open up that box and it's got 1,000 Legos that instead of being one shape are now 20 different shapes. How much stuff can you build with that? Well, the answer is a lot, right? Probably anything you imagine you could build with 20 different shapes of Legos. And so that's where proteins come in. Just like carbohydrates, just by lipids, they're built of repeating units. But in this case, the monomers are amino acids. And those amino acids have 20 different forms, which means we can combine, we can make proteins in a variety of shapes, and they can do tons of different things. Here's a selection of some different types of proteins from your book. Notice all these different things the proteins can do. And so, really, the functional molecules that get a lot of stuff done in your body are going to be built of proteins, because proteins can perform a variety of functions. Protein structure is divided into several levels. It starts off with a sequence of amino acids, and then depending on their order, it eventually coils up into this three-dimensional shape, and we can make several different chains, combine them together, and all sorts of cool stuff. These bases will be determined by a nucleic acid sequence on DNA. We will talk a bit more about that when we talk about cells and genetics in the next upcoming chapters. Nucleic acids, I already mentioned them. They're polymers composed of nucleotides. DNA, known as deoxyribose nucleic acid, has a sugar group, contains a sugar, a phosphate, and one of 4 nitrogen-containing bases. So DNA, deoxyribose nucleic acid. Those 4 possible bases are A's, T's, G's, and C's. You will see a great many of these in your upcoming chapters, particularly those on genetics. DNA, by putting these in different orders, conveys information or contains information. So, for instance, like letters in sentences in a book you're reading, they combine to make meaning. The order of arranging A, T's, G's, and C's in DNA contains meaning and instructions for building proteins. Remember, proteins were awesome, which ultimately are parts of almost everything you do in your body because of their diversity. And so you'll see this structure in the future some more. The double helix, A's bind with T's, A's bind with T's, C's bind with G's. And there's also a second type of nucleic acid you learn about known as RNA. And so this is kind of a summary of the biological molecules. All right, so we talked about a lot of different stuff today involving chemistry and its importance to the biological world. And if you remember back at the beginning of class, we started with that hierarchy where we talked about how atoms make up molecules. Molecules then make up larger things. And in this case, molecules can be combined to make organelles. Organelles, the word sounds like organs, are really like the organs of cells. And so next week, what we'll do is we will move on to talking about another group of small things in biology, which is cells. Chemicals are super small, moving up a little bit to slightly bigger things with cells. And I hope to see you next week for our lecture on cells.